from the atrium. It can't get through this fibrous septum. It can only slowly travel through the AV node. Very slowly. It takes 0.1 of a second. So that means that depolarization of the atrium, then contraction, then there's a 0.1 second break while the depolarization wave moves through, and then it continues to propagate. You have a flat line while it's propagating through. So, once that's happened, once the action potential or the depolarization event has moved through, this AV node branches off like this. Okay. And the left branch, now this is called the bundle of His, okay? The bundle of His branches into the left and right Purkinje fibers. But the bundle of His, you'll see the left branch branches off like that, okay? So that means once this depolarization event has reached the AV node, flat line, it then propagates through these fibers. Now these fibers are really fast, okay? Very, very fast. And as the depolarization wave spreads down, as it goes down, you can see that it goes down like that, but also down the left bundle and starts to fire up like that. All these branches firing up. So it depolarizes the septum between the left and right ventricle, like that, in that direction. You can see in that direction. That means positive stuff is moving in that direction. So that means, look at the lead. Is this depolarization event going towards the lead or away from the lead? Overall, away from the lead, because as it moves down, these ones are going up like that. Okay? So that means what you get is depolarization away from the lead, a deflection, a bump down. Now, this deflection isn't nice and rounded like that because these fibers are very fast, and because it's not a thick tissue, it doesn't last very long, you get a bump down like that. Okay? Alright, once the depolarization event has reached towards the apex, these bundle branches move through and turn into Purkinje fibers that move up like that. And these Purkinje fibers branch off like that. Okay, so that means it's depolarized down, we get a deflection because it's going away from the lead, then it reaches here. And what you'll find is it starts to depolarize like that. So have a look. Is that overall in the direction of the lead or away? Now you may think, well, this part's in the direction of the lead, so the left ventricle is, but the right ventricle is going away from the lead. So you may go, I don't know what direction this is happening. Well, remember, the left ventricular wall is about 3 times thicker than the right. So what's happening in the left ventricular wall is going to overpower what you read on the ECG to what's happening on the right. So what's happening over here? Predominantly, the depolarization event, which is positive stuff, is going in the direction of the lead. And because the tissue is thick and they're very fast fibers,
So,
you have a very high bump up. Like that.
yo,
Okay?
Horse
Now,
edits
it's
and
reached
her
to the,
doodles?
what we call towards the base.
Course
So remember, that's the base
1022?
of the apex. Once this
What?
depolarization event
You?
is reached towards the base of the ventricles, the depolarization event moves up.
Yeah.
So this is the part
You?
we're looking at now.
What do you mean by
You
you?
can see it moves
That's
up
supposed to
and
be like
away.
a...
So we have depolarization going away
Anyways.
from the lead. It's not a very big bit of tissue and they're fast fibers, so we get another
No, I get the...
deflection, another bump down. Cool. All right. What's the next part? We have now just depolarized the entire heart. It's all positive now, right? And
No,
it's all contracted.
the best thing to
So
do.
sodium comes
It's
in,
like,
calcium contracts, sodium
say,
comes
real
in, calcium
talk.
contract. Now what do we need to do? Well, we
That's
need to repolarize it.
a lot.
Okay?
You already
That's
messed
where
it
the
up.
that's where the
Crazy.
potassium leaves the cell to make it negative again.
Study and then...
inside. Okay? So that means we need to make all this negative again. Now what you need to realize
Talking
is
out
this.
loud there, Lincoln.
Even though depolarization
Listen to some
started
audio in the
here
background.
and moved like that, and then went to the AV
I
node,
like
moved
plan out my
like
day.
that, and then like that.
Oh, you heard me,
Repolarization
player
does not
slayer.
start
You heard me,
where
bud.
depolarization started. Repolarization starts where depolarization finished, up here. Okay? Which means now repolarization goes in this direction. Okay? Repolarization goes in this direction. So, as it goes down, all you need to do is look at the opposite of the dipole. It goes in the opposite direction to dipole. And you saw dipole predominantly go in that direction for that big bump. So repolarization goes in the opposite direction. Okay? And that's making it negative again. So you can see this is happening away from the lead. Right? So we have a repolarization event going away from the lead. What do we get?
Bro, scared, bro. Bro, scared. You walked off the screen, bro. You're scared, bro. You're scared, bro.
And this is your ECG. That's going to reset now. That's going to go back to negative, and then the next cycle can happen. So this is one cardiac cycle. This is one, boom, boom, resets, boom, boom, resets, boom, boom, resets, boom, boom. Okay? So this is what we call just a general ECG readout. And you can see it's basically an ECG readout from the V5 or lead 2 cable. So, what does that mean overall? It means this. If we... Hi everyone, Dr. Mike here. Today I want to talk about neuroplasticity. Neuroplasticity is the brain's ability to reorganize itself, make new neural connections. So how does this work? Well, I want to provide an example that I think highlights it quite nicely. But what I first need to show you is the brain itself. So I've drawn up the brain. This is the front of the brain, this is the back of the brain. And you can see the brain has all those grooves. Now, if it dips in, that's called a sulcus. If it bumps out, that's called a gyrus. Now, what you'll find is that right in the middle of the brain here is a sulcus that separates the front aspects from the more posterior aspects of the brain, and this sulcus is called the central sulcus. Now, interestingly, in front of the central sulcus is where we control motor movement, and behind the central sulcus is where we feel sensations. I want to focus on the sensory area behind the central sulcus. It's actually called the somatosensory cortex. Now the cortex is the very outside 1 to 2 millimeters of the brain. So if I were to get a knife and I were to cut down the central sulcus, discard the front of the brain, and have a look from the front of what we can see once we've cut it, we'll see something that looks a little bit like this on one side of the brain itself. Now let's talk about feeling sensation. Let's just say somebody just touched my index finger. That signal goes from my finger down my arm into my spinal cord, up to my brainstem, crosses the other side at the medulla, and then goes up to the thalamus. The thalamus then projects it to the somatosensory cortex, specifically the area of the cortex that deals with me feeling things with my finger, which is actually located right here on the most lateral aspect. All right, now, as you can see, if there's a part of my somatosensory cortex dedicated to my finger, there must be a part dedicated to other body parts, and that's what you can actually see here. There is a map of our body on our brain, and as you can see, it's not the same map as what our body actually exists in. What you see is this. The most medial side, you're going to have the genitals, then the foot, then the leg, then the knee, then the neck, then the arm, then the hand, then the face, including the eyes, nose, lips, tongue, and voice box. That means that, for example, when I touch my finger, I know that someone or something is touching my finger once the signal gets to this part of my cortex. It also means if I were to go into this person's brain with an electrode and stimulate that area, I would think, or that individual would think, that they are having their finger touched, right? All right. What happens when somebody loses their arm? Or let's just say they lose their hand, for example. Well, it means that all the sensory neurons for the hand now no longer exist. But all the pathways still exist all the way up into the brain, including the map for the hand on the brain. Do you think this map on the brain for the hand still receives signals? The answer is no. It's not receiving any signals. And the brain gets hungry for signals. It loves receiving signals. So what happens is the neurons that are present here get a bit bored. They start to have a conversation with the neurons surrounding it. Now, the neurons surrounding those for the hand are those of the face. And so what that means is a conversation can now occur, and in some individuals, you can tell them who have lost their hand to close their eyes, and you rub down the side of their face and ask them, what are you feeling? And they say, well, I feel you rubbing my cheek, but I also feel you rubbing my index finger, my middle finger, my ring finger, and my pinky finger. Why? Because the neurons have started conversation, and this is neuroplasticity. I'm Dr. Mike. Hi everyone, Dr. Mike here. There are 100 trillion cells within your body. A red blood cell can actually be around about 10 micrometers in diameter, not very big, one of the smallest cells. You can have a sperm cell, which is around about 50 micrometers in diameter, still not very big, but you can have a nerve cell that can be around about 1 meter long. So cells can vary quite significantly. When you have a group of cells working together towards a primary function, these cells combined form a tissue, and we know that various tissues, such as connective tissue, muscle tissue, nerve tissue, epithelial tissue, when they come together, they form an organ or organ system, and we are an accumulation of organs and organ systems. So the cell is the basic unit of life. Now, what is similar from one cell to another? Well, what I've drawn up behind me are some of the similarities between the cells of our body. First thing you can probably identify are two major subcellular structures, one of which is the nucleus itself. The nucleus houses our genetic material. This is our DNA and our RNA. Then you can see the rest of it is what we call the cytoplasm. The cytoplasm is all the fluid within the cell and also all the subcellular structures that we call organelles. Okay, if we start again with that nucleus, you can see that we've got a nucleolus. A nucleolus is an accumulation of RNA and some proteins, and the nucleus, which houses our DNA in the form of chromosomes. This is a condensed form of our DNA. As we move outside of the nucleus, you can see that there is this structure here which looks like it's continuous with the membrane of the nucleus, which we call the rough endoplasmic reticulum. There's actually two endoplasmic reticulums, a rough and a smooth. Now, the rough is called rough because you can see it's studded by all these red dots, which we call ribosomes. Now, the function of a ribosome is to take RNA that has just come out of the nucleus and read it and turn it into amino acids. These amino acids fold into proteins. So ribosomes effectively turn DNA and RNA into functional proteins. And you can see those attached to the rough endoplasmic reticulum mean that the proteins they make are embedded in the rough endoplasmic reticulum. They are played around with a little bit, they're altered, their structure is altered a little bit, and then what they do is they're passed on to something called the Golgi apparatus. You can see that this green structure here is the Golgi apparatus, again, a membrane-bound intracellular organelle. What the Golgi apparatus does is it slightly alters the packaged material that was given to it from the rough endoplasmic reticulum. For example, some of the changes it can make is it can add some things to the ends of it, like little flags that tell this protein where to go and what to do once it gets pushed out of the cell. So that means the Golgi apparatus creates little vesicles, these little bubbles here, that fuse with the wall of the cell, and it releases its components, and these components are predominantly proteins that are made by the rough endoplasmic reticulum. They're slightly altered and they can travel elsewhere throughout the body. So that means the proteins made by the rough endoplasmic reticulum are destined to be exported out of the cell. You can think of it as though this is the factory, the rough endoplasmic reticulum, making the product. The Golgi apparatus is like the post office, putting the stamps on it, determining where it goes. Now, the smooth endoplasmic reticulum doesn't have ribosomes on it, which means it doesn't make proteins, but what it does make are lipids. These are fats. It also plays a really important role in detoxification. That means it can take drugs and other certain compounds and detoxify them, break them down. So one of the organs of our body that does this as its primary role is the liver, which means therefore the liver has huge amounts of smooth endoplasmic reticulum. Now, some other important structures include the mitochondria. The mitochondria create energy in the form of ATP. It can actually take glucose, which is the basic monomer of sugar and oxygen, and together it can create ATP. One glucose molecule can create between 32 and 36 ATP molecules. So mitochondria create energy. It also has its own DNA, and that means it can make, through binary fission, split itself apart and make its own copies. Now, inside the cell, around about 75 to 80% of it is water, but the second most abundant substance are proteins. And you can see you can have structural or functional. Structural proteins, such as these microtubules, are basically like the scaffolding of the cell that holds it together, but it can also play an important role when this cell wants to make a daughter or another copy of itself. These microtubules are used to help split it apart. All right, we've also got these two important substances here. This is a lysosome and a peroxisome. Now, a lysosome is actually a little vesicle that's budded off from the Golgi apparatus, and the lysosome plays a really important role in breaking substances down. It degrades things. It contains enzymes called hydrolases. Hydrolases means it uses water to split things apart. So it can take proteins and split them apart into amino acids. It can take glycogen, which is complex sugars, and split it apart into glucose. It can also take fats and split it apart as well. Another subcellular structure here is a peroxisome. Now, a peroxisome is very similar to a lysosome. It breaks things apart, but it didn't come from the Golgi apparatus, okay? And what the peroxisome does is it actually breaks things apart using oxygen. So what it does is it oxidizes substances. So it can actually take oxygen and hydrogen and create something called hydrogen peroxide and use that to split things apart. Specifically, it splits apart fats, long-chain fatty acids. It can break apart using hydrogen peroxide. Now, some other important things that you should note is that when we look at the cell, it's surrounded by a phospholipid bilayer. That means there's a lot of fats embedded in there, and we know that fats only let fatty things in and out, or at least things that can dissolve in fats. That allows for us to separate out what's happening inside the cell from what's happening outside the cell. So this is a very quick work through with the cell and some of the subcellular structures that we call organelles. Hi everyone, Dr. Mike here. In this video, we're going to talk about connective tissue. Now remember, connective tissue is one of the 4 tissue types of the body. It's the most abundant and also the most widely distributed. Now, some connective tissues are solid, like bones, some are semi-solid, like cartilage, and some are fluid, like blood. So what makes connective tissue connective tissue? Well, all connective tissues have cells, gels, and fibers, and what makes one solid or semi-solid or liquid has to do with its composition of different types of cells, gels, and fibers. So for example, if we start with cells, you'll find that there's many different cell types that fit within the connective tissue umbrella. For example, fibroblasts. Fibroblasts create skin. Chondroblasts create cartilage. Adipocytes, fat cells. Osteoblasts, bone cells. Now these are just some examples of the cells within connective tissue. If you see the suffix blast, it means to build. And these are the early immature cells of that connective tissue. So fibroblasts build skin cells, osteoblasts build bone cells, for example. If you see the suffix cyte, it's a mature cell. Let's move on and look at the gels. So the gels within connective tissue are predominantly water, but it's got some substances dissolved in the water. Some of these substances include the GAGs, which are the glycosaminoglycans, the proteoglycans, and the glycoproteins. So these are basically sugars and proteins together, and again, different types of connective tissues have different quantities of these types of gels. Now,
You see in the closet, Maximus?
Collagen, elastic, and reticular. The way I like to think of these fibers is collagen is like steel metal rods, very strong. Elastic are like rubber bands, very stretchy. And reticular is like cotton wool, like a mesh-like framework. Okay? Now, what you'll find is when we have the gels and the fibers put together in varying concentration of each of its substances, that then is going to result in either being a solid, a semi-solid, or a liquid. So for example, if there's a lot of collagen fibers, it's going to be very strong and hard bone, for example. A lot of elastic fibers, it's going to be very stretchy, like blood vessels. If it's got a lot of reticular fibers, it's going to form like a mesh-like framework, like you're going to find in the lymphatic vessels. the jelly, and you can dissolve these fibers inside of that jelly
Connective
to change the
tissue.
consistency.
That's
Now,
old school.
When we look at the various
That's old
connective
school
tissue
stuff, man.
types, you've got connective tissue proper, and its primary role is to bind things together. You've got supporting connective tissue, its prime protection and structure, maintaining structure. And you've got fluid connective tissue, which is blood basically, and that's there to transport stuff. That's going to be gases, nutrients, and waste. So if we first look at the connective tissue proper, there's two types. There's dense connective tissue and loose connective tissue. Now, dense and loose has to do with what's inside. So dense connective tissue is going to be quite dense. There's a lot of fibers, it's predominantly a lot of collagen fibers.